بسم الله الرحمن الرحيم. أعزائي الطلبة الصف الثاني، سنبدأ اليوم بعد ما أكمل كل الأستاذ الأخ دكتور أمجد محاضراته. سأستمر معكم بحدود 10 محاضرات حول مادة الإندوكراين سيستم. فبعد التوكل على الله هذه المحاضرات طبعا أي إشكال في أي موضوع يرجو التواصل من خلال الكلاس روم، وإن شاء الله تكون مفهومة لديكم. Introduction insight into the hormone action. The endocrine system is a very crucial biological system that works to regulate a wide range of our body biological processes that are essential to maintain life. It is composed of a number of glands that release different kinds of biochemical compounds known as hormone directly into blood stream under the control of many mechanisms like the neuro-endocrine coordination. Among these glands are pituitary gland, thyroid and parathyroid, suprarenal adrenal gland, GIT gland, pancreatic gland, which include different type of cell, which is called the alpha, beta, gamma, and F cell of the islet of Langerhanz. And there is the gonad with hormone. The hormones can be classified by their chemical composition, solubility, location of their receptors, and the nature of the signal that mediate hormone action. According to the last 2 points, hormone can be classified into 2 classes. First, the hormone that bind to intracellular receptor, example, thyroid hormone, mineralocorticoid, glucocorticoid, and sex hormone, and retinoic acid. Second, the hormone that bind to the cell surface receptor. The second messenger is cyclic AMP. Example of that hormone that work in this mechanism include catecholamine, except alpha-1 adrenergic, FSH, LH, PTH, TSH. B, the second messenger is cyclic GMP, example in natriuretic factor or peptide. C, the second messenger is calcium or phosphodiesterases, inositol, or both, example in alpha-1 adrenergic catecholamine, acetylcholamine, gastrin, and etc. The last group is the second messenger is the kinase or the phosphate cascade, example insulin, insulin-like growth factor, IGF-1 and IGF-2, prolactin, and growth. For hormones of group 1, they are lipophilic, that mean water insoluble. They thus need carrier protein in the plasma. They possess a longer half-life. Their intracellular receptor could be either systolic or nuclear, and the ligand receptor complex, in this case the hormone receptor complex, is the second. On the other hand, hormone in group 2 are water soluble, first, have a shorter half-life, that is mean in minute, and bind to the cell surface membrane and can't pass to inside, but their action is mediated through an intermediary molecule called second messenger. The hormone by itself is the first messenger. When the hormone of group 1 is carried till the cell surface, it diffuses within the membrane by its lipophilic property, but ultimately bind to its specific high affinity receptors, whether the nucleus or in the cytoplasm. The hormone receptor complex undergo a temperature and salt-dependent activation reaction that result in size, configuration, and surface charge changes that enable the complex to bind to specific region of the DNA, which is called the hormone response element, in abbreviation HRE. In this way, the hormone receptor complex activate or inactivate a particular gene in the DNA sequence, resulting into specific messenger RNA that would leave to cytoplasmic and being translated into specific protein which express the primary messenger, the hormone. Example, calcitriol 125 or 125 dihydroxycalciferol or the active vitamin D enhance the synthesis of a specific calcium binding protein in the intestinal mucosal cell and thus promote calcium absorption, as seen in this diagram. While in group 2, that is the peptide hormone, this group contains the vast majority of the hormone. These are water-soluble. They bind to the receptor in the plasma membrane, but utilize the following intracellular secondary messenger. The first and the common one is the cyclic AMP. Cyclic AMP is a nucleotide that is derived from ATP through the activity of an enzyme, adenylate cyclase, binding of the hormone that utilize this mechanism to their specific receptor will result in their activation or suppression of this enzyme, and thus in either increase or decrease intracellular level of cyclic AMP. However, this process of activation or suppression is also a tissue-dependent process. Adenyl cyclase is present on the inner surface of the cell plasma membrane, and it is regulated by at least 2 GTP, that is guanylyl triphosphate dependent protein, which are either GS and GI. When this enzyme is stimulated, it catalyzes the conversion of adenine triphosphate into cyclic AMP in the presence of magnesium. In the eukaryotic cell, 4 molecules of cyclic AMP bind to a specific protein kinase, which is responsible for protein phosphorylation. This binding activates this protein kinase, and active protein kinase will utilize an ATP molecule in the presence of magnesium to convert protein to phosphoprotein, which would then exert their biological effect. Action caused by hormone that increase cyclic AMP concentration can be terminated by phosphodiesterases. This hydrolytic enzyme ensure a rapid turnover of the signal, cyclic AMP, and hence rapid termination of the biological process. As you see in this diagram, inactive protein kinase will share into these different 4 cyclic AMP, which result in activation of the ATP in the presence of magnesium. It converted to cyclic AMP, and this will activate the protein kinase, and the protein will be activated in the presence of AMP. It will be converted to the phosphoprotein, and this will exert the physiological effect. And by the presence of enzyme, which is called the phosphodiesterase, it will convert cyclic AMP again to the AMP. to terminate the action of cyclic AMP. And you see this is the structure of the cyclic AMP. You can see 5 connected to the 3 carbon atom to make cyclization of this TP to become cyclic AMP. What is more about the role of calcium in hormone action? Ionized calcium is a well-known regulator of several biological processes, including muscle contraction, nerve conduction, collecting mechanism, bone formation, enzyme activity, hormone action, and the second messenger. Class 2C on the first classification, on the second or third class slide, hormone enhance membrane permeability to calcium ion and thereby increase calcium influx cellularly. This calcium dependent regulator or regulatory protein is now referred to as a calmodulin, which is similar in structure and function to muscle protein troponin C. It has 4 binding sites to calcium. Once these sites become fully occupied by calcium, this will produce a profound conformational changes in the calmodulin molecule. These changes will ensure an activation process of calmodulin. Such activation is similar to binding of cyclic AMP to protein kinase and subsequent activation of the latter. Active calmodulin is particularly involved in regulating various kinases and enzymes of cyclic nucleotide generation and degradation. Example, pyruvate kinase, adenyl cyclase, glycogen synthetase, other. What is remaining is how the role of the phosphatidyl inositol metabolism in calcium-dependent hormone action. When the cell surface membrane are occupied by their specific hormone, such as ADH antidiuretic hormone, acetylcholine, or alpha-1 catecholamine, they become a potent activator for phospholipase C, which is an enzyme in the plasma membrane of the target cell. Activation of the phospholipase C is occupied by the effect of unique protein G protein, which would together result in increased calcium influx to intracellular space on one hand, and to hydrolyze of the phosphatidyl inositol biphosphate, or what we call PIP2, into diacyl glycerol and inositol triphosphate, or IP3. The other hand, diacyl glycerol activate a protein kinase, a process which is need calcium for activation. Active protein kinase will change a protein in the cell cytosol into phosphoprotein, which will then exert their biological effect. Inositol phosphate 3 act on the intracellular reservoir of the calcium ion that present in the endoplasmic reticulum and mitochondria to release further calcium ion. In this way, the hydrolysis of phosphatidyl inositol molecule result in both activation of protein kinases and increase the concentration of intracellular calcium. The high level of calcium inside the cell membrane, calmodulin, at its specific binding site, the calcium calmodulin complex can activate both specific and multifunctional calmodulin kinases within also phosphorylate protein, as shown in this diagram, all the mechanism. The last mechanism shared in the 4 group is the insulin and insulin-like growth factor, in addition to other hormones like prolactin and growth hormone, act through a cascade of kinases and phosphates, specific receptor of these hormones, which contain intrinsic tyrosin kinase. Activation of this enzyme lead to activation of number of cytoplasmic protein tyrosin kinase, which will phosphorylate one or more of the cytoplasmic peptide segment, which will then get the way to nucleus and bind to specific region of the DNA and activate a translation